welcome to the New York State IPM Academic Seminar Series designed to increase awareness of new research and techniques that advance IPM in all types of pest management settings. We typically have about a 40-minute seminar, 10 minutes of questions, and then 10 minutes for an informal meet and greet with our speaker. I do want to pop into the chat a couple of links here to our events page, the New York State IPM website, as well as our YouTube channel, where this and other seminars are typically posted. And now I would like to turn it over to Brian Eschenauer, Associate Director of Program-Wide IPM, who will introduce our speaker.
Thanks, Brian. I'm happy to introduce Julie Lockwood today. I have to say that the environmental techniques I was aware of were for the use of detection of aquatic invasive species, and I learned of its application for terrestrial invasive species through Julie's work with spotted lanternfly. Looking forward to hearing all about her work with eDNA. But before we get started, a little bit of background on Julie. Professor Lockwood is the interim director of Rutgers Institute of Climate and Energy and professor of ecology at Rutgers University. Her research centers on the prevention and management of invasive species, the socio-ecological dynamics of the wildlife trade, and the impacts of climate change and energy and clean energy on biodiversity. She is a fellow of the Ecological Society of America and of the American Association for the Advancement of Science. Hey, we're happy to have you here today, Julie.
Well, thank you. I had to find my mute button. I, of course, can't see any of you, and so hopefully someone will throw something in the chat if there's something I should address along the way. But thank you very much for inviting me in for today's talk. I'm happy to share this work. I've been loving doing this and sort of thinking about how DNA applies in a lot of different contexts from someone who was trained as a traditional field biologist, not as a molecular ecologist, and realized the power of sort of these approaches and this kind of new molecular tech, if you will, for solving a lot of the problems that I encountered when I was more doing field work related things, where you're just spending hours and hours and hours in the field trying to find rare things. And it was very disheartening, honestly. And so when these came along, I'm like, we have to try to do this. So I've kind of rejiggered my lab and ramped up what we do. So I'll just preface that by, if you think that eDNA sounds crazy, so did I. And then I did not come at this as a molecular ecologist. I came at it through a field ecologist who's interested in invasions and species extinctions. Okay, so with that introduction, the title is, of course, Finding a Needle in a Haystack and How We Can Use eDNA in Detecting and Managing Pest Insects. Whoops. There we go. So just a little bit of overview of where we're going. I'll talk a little bit about the importance of detection of low abundance populations, the needle and the haystack, especially related to, well, really invasive species management in general, but in this case, I'll concentrate on insect pests. And then address, how does eDNA kind of help us in terms of the detection of low abundance populations? And then, eDNA to the rescue, a big question mark. And then I'll go in the background of how eDNA even works in the context of terrestrial insect species, and then what we've sort of, the methods and things we've kind of innovated and tried to push. And then I'll show you some of our preliminary results, and finally have a little bit on kind of what's on the horizon. There's still a lot of questions about moving this technology into pest management, honestly. So what components of pest management are akin to finding a needle in a haystack? I would say any part that requires detecting individuals of a pest species when they're very rare in a landscape. That would include a whole slew of things in my experience. That includes biosecurity surveillance, stop the spread campaigns, any time when the economic damage of a pest insect, in this case, is incurred at very low abundances. So it's sort of a highly impactful species, and you can't let it, like the the economic threshold at which you might want to apply a management tool is very low, so it doesn't take much and you're going to have a problem. If early seasons or the emergence time, basically, if you can detect them at that moment when they're kind of rare and just popping out, does that save you money or guide management? That's another situation. Or to confirm local control or eradication, that's something we're doing regularly, actually, in aquatic eDNA. And it's a very good tool for that as well. And then finally, detecting reinvasion events. So basically, anytime you are confronted with a problem of finding the target species that you're interested in when it's really rare, when it's not very common on the landscape or in the sampled area that you're looking for, that's when you're really trying to find a needle in a haystack. You're trying to find that one individual that triggers some sort of management response. And that individual's hard to find. So this brings up two definitions that I will mention pretty regularly through the talk. So I'll take a minute to define what they are. One is detectability, and the other one is probability of detection. So detectability is just the ease with which individuals of a past species, in this case, can be noticed or discovered at a given location. So this one has so much to do with kind of the life history of the species, its behavior, our interaction or where it's at relative to where we're at. It's basically how likely are you to run across that individual when you're walking in the woods or you're walking along a vineyard or even by your house. Sometimes that's very obvious. This picture right here is of a coconut rhinoceros beetle. So that's a past species in the Pacific, and we've been working on that one, and it's a pretty big beetle. And so when you see it, when it's there, you notice it. It's a big beetle. So its detectability when it's around is really quite high. Now, in this case, the reason we got pulled into this one is they're nocturnal and they spend almost all their time at the canopy of palm trees. So even though they're very detectable, in some sense, they're very not detectable because of behavioral and other issues. So another piece of that, though, sort of the follow-on is probability of detection. So this is a statistical metric that describes how often a species is detected when it's present in a location. So detectability plays a role in that. So if you have a very detectable species, you should have a high probability of detection. But there are other things that go into that probability and what that looks like. So in particular, I think most of us understand that when you want to find a species in the landscape, a pest species, there are really three things that matter. One is, what is its underlying abundance, which is a constant theme throughout this talk, which you've seen so far. This particular graph is a great graph because it illustrates my point. It comes from birds, but if anybody knows of one that comes from insects, then pass it on and I'll throw that into my next talk. But the basic picture here is that detection rate, or the probability of detection, goes up as the abundance of the target species goes up. So all those little circles are different species with different abundance and different detection rates. And that kind of increasing curve indicates that at really low abundances, right down here, detection is very rare. And it gets better, right? But for a lot of the things that I'm going to talk about, this is the most critical point that you need to find the pest insect. And I'll explain why that's true. So when they're rare, you don't see them very often, which means they have a very low probability of detection. The other thing that happens is the way that you look for them matters, right? So trap efficiency, especially in terms of pest insects, we have a whole long, nice history in terms of research in this area. Is it how do you design traps to kind of uncover those rare individuals that are out there? How do you create? Do you load them with pheromones? Which pheromones work? How far do they work? Where do you hang them? Where do you put them? All those things have to do with detectability. So your probability of detection goes up when you have a really good trap that has high trapping efficiency. And then the final thing, which I think we all know, is that the sampling intensity or the sampling effort really matters. So if you spend a whole lot of time trying to find that rare thing on the landscape, you're much more likely to find it. So your probability of detection goes up with sampling effort. So those three things we know really matter, and I'll head on these ideas kind of over and over again. But I'm gonna concentrate on this problem of finding the needle in the haystack. So how do we do this? How do we efficiently find individuals of a pest species when they're very rare, which is a hard job? Why are we so concerned about it? Like, why am I obsessed over that? I mean, this is where maybe all that my prior field life kind of comes into play. In terms of forest pests, at least, there's some great work by Patrick Tobin and some of his colleagues that show that the sooner you catch an invasion of a pest insect, the much more likely, the higher the chances are that you can achieve true eradication. So that top graph on the left is just it's a binomial plot. So the eradication that they tracked was either successful or not. And then the y-axis down at the x-axis at the bottom is the area that species occupied. So it's very rare on the far left, and it gets less and less rare up to common towards the right. So you have really high probability of success for true eradication when that pest insect is really only maybe occupying 100 kilometers square or less. After that point, that line tends to bend down, which means that it's just a lot of diminishing returns at that point. Your chances of successful eradicating is low. And then the bottom part has the same kind of idea, but the cost. So the cost goes up. This is a log plot on the bottom, so it looks nice and linear, but it's actually exponential in real, not log plot land. And what this suggests is that the costs of those eradications is also low, and that cost goes exponential as the area basically more abundant that pest species is. So this is a huge premium on finding that pest insect on the landscape when it's very rare, because what that means is that you can have a high chance of eradication for not a lot of money. But as soon as you break out of a certain threshold, your chances of success go down, and you'll spend a way lot more money. The other side of this is just they're looking at detectability and method of detection, and where the successful eradications are black and the failures are in gray. So what you see is that when you have a highly detectable species, your chances of eradication are quite high. And if you have a method of detection, a sampling process that you can actively increase or decrease effort, mostly increase effort in this case. If you can increase effort, then you also have a good chance of finding them and eradicating them. But if you're waiting for passive or reports maybe from citizens or any kind of passive method of detecting them, your chances are very low. So this is, to me, like this is true, I would say, just from anecdotal and other evidence. This is true across the board with every invasive species I've ever looked at. So these relationships are universal. So this puts a lot of premium if you're worried about invasive species and you think you have a prayer of eradicating, then you have to find them when they're very rare. The other two times I see it come up is in biocontrol, both in terms of confirming that either your biocontrol worked and that the target of the biocontrol, the pest species, has either dropped to zero or is at some sort of lower threshold. That low threshold often is very small, which means that now we're back to rarity land again, where you're trying to figure out what's going on with that species. Is it still there or not? And if it is, where is it? So you're trying to find that needle in a haystack again. The other part of that is when you release a biocontrol agent, you're starting at a very small population size. So you're starting at low abundance, and then you're asking, I wonder if it worked. So you're then again trying very hard to figure out, okay, are they still there? Do I need to release again? How many do I need to release? Do I need to release in one other area close by? So now here again, the premium is finding the needle in the haystack. So at both ends of biocontrol, you kind of, this premium on finding individuals in the rare in the landscape is huge. And then the final one is decreasing the time between invasion, sort of the establishment of an invasive species, and then when we have an initial detection. that lag between when they first showed up, like in biological reality, they showed up versus when we find them, is also, for everything I've just said, is incredibly important. So now we're back to, how do you know? Now you're not even entirely sure what you're looking for, but you need to find things that are new into the area. You need to find them soon. This is just a plot. This is a dendrochronological sort of reconstruction of the invasion history of emerald ash borer out of Michigan. So this is all data that's kind of back forecast or backcasted based on tree rings. And this, for those of you who know the emerald ash borer story, you know that it was established and growing in the late 1990s, but we didn't get an initial detection until the early 2000s. That's a lot of missed opportunity in that case, because if we had found it a little bit earlier, we could have probably been in that nice, cheap eradication land. And now we're, of course, far from it. So we need to find individuals of pest species when they are very rare in the sampling area or on the landscape, but this is precisely when it's most difficult to find them. It's like, perfect, that's perfect, great. Now what are we supposed to do? So, when the truth is out there, we have to kind of find it. And then there's a high premium on figuring out what is going on in any one of these cases in terms of pest management. So now, we get to this, is environmental DNA to the rescue, big question marks. And then, environmental DNA girl there, I guess. So now I'll kind of dig into that possibility in more depth. And so the whole idea of environmental DNA is that, the promise of it basically is that you can detect the presence of species that are either there now or were there very recently through collection of environmental samples. And so it's a sort of sight unseen detection. And you don't have to capture them, you don't have to hold them, you don't have to kill them, you don't have to do any of that. You just need to find their DNA. So let me give you the background on that. So environmental DNA, the definition of that is that it's genetic material that is shed by living organisms into the environment and can be sampled through collection of bulk materials, usually either soil or water, or from air, or from surfaces. We do it on natural surfaces. Again, it's sight unseen detection. As Brian indicated, most people are familiar with eDNA because of its use in aquatic systems. That's where it was originally kind of developed. That's where the term eDNA first came from, was in this context of water. The mechanism there seems clear, right? I mean, fish are swimming around, they're kind of slimy anyway, and all that slime goes different places. They shed cells, they excrete things, and all that has their DNA in it, and it gets suspended in the water column. And it just sort of holds there for quite a long time. So if you can grab the water and look for DNA, their DNA in there, then you can figure out what was living in that water. So that was pretty cool. I remember reading about that when it first came out. I'm like, wish we had something like that for terrestrial systems, but I mean, how would that even work? Because part of what's happening in environmental DNA in water is the DNA is getting suspended in the water and kind of floating around in the water. So it's pretty clear what you're supposed to sample. For terrestrial systems, it's like, what are you supposed to sample? So one of the things that we realized from doing other work is that really forensic DNA is also environmental DNA. So in forensic world and crime world, then they call it touch DNA sometimes. But every time you touch a surface and anything that touches a surface as a living organism will deposit some of their DNA on it. So in forensics land, they have a little swab and they'll go in and, like the detective scene in CSI, and they're sort of swabbing that surface and trying to find the DNA of the killer. We thought, okay, that probably works, but we have to think like an insect. And so where are they going to leave their DNA, and can we get it? Right. So if we can get it, and we can get it a little bit more efficiently than taking a little forensic swab and trying to swab the world, then maybe it'll work. So that's the genesis behind this. Most of what I'm going to talk to you today about is our sort of exploration of taking that forensic DNA idea, that touch DNA, and making it work for terrestrial species, in this case, mostly insects. before I do that, this is a common question, is like, how do you know from the DNA that that species is there? What is it about the DNA that's so diagnostic that you know that that species was there? So in this case, what's happening is you're picking apart any segment of DNA, and you're doing some bioinformatics magic, basically, that says this sequence of ACTs and Gs on this part of the genome, and if we get that sequence, it's unique to that species. And so there's a lot of background. You have to establish that kind of in the lab. And again, through some just lab testing of that, but also some bioinformatics to associate one segment of DNA in one region to one species. And so that's what you're looking for. For environmental DNA, that segment is really pretty short. It's usually 100, 200 base pairs. The reason for that is because as soon as DNA leaves the living person or living individual, it starts to degrade, it starts to pull apart. So UV degrades it and basically breaks it into smaller and smaller chunks, and so do microbes. So the DNA that you're going to pick up in a water sample or on a leaf or in the soil is going to be short because it just immediately starts to fall apart into smaller and smaller fragments. So for environmental DNA, you're taking a small fragment of DNA with a particular base pair sequence, and you're making sure that that base pair sequence is unique to the species that you're looking for. So there's a lot of little background work that often people don't talk about, but that's what has to happen for an eDNA process to work. That's a little bit of the background. Okay, so then we come around to like, if I have to think like an insect and think about where they're depositing DNA, where do they deposit their DNA? So we've come up with, of course, a lot of different solutions. Every species is different. One of the wonderful things about dealing with insects and arthropods more generally is they're so cool and they do so many different things. But the pest insects that we've looked at so far included Capra beetle, Asian longhorn beetle. We haven't yet done anything with strawberry pests, but that's a possibility, and then spotted lanternfly. So each one of these species has, of course, its own life history and does its own things and uses different surfaces. And you have to think like that insect to figure out where you're supposed to go searching for their DNA. So for Capra beetle, for example, we're working mostly with USDA APHIS on infestation of bulk grains like rice. So in that case, what's happening is they kind of chewing their way through all that chew juice basically has their DNA in it. They can defecate in there, but they also leave, as they molt, they leave their exoskeleton, which can get ground up and everything else. And so there's a lot of DNA in that sort of bulk container. Some species like Asian longhorn beetle will leave a frass. This is like an obvious frass situation, but really we don't need much. I'll show you in a minute. You don't need much DNA for these things to work. So we wouldn't need nearly that much frass to be able to find an Asian longhorn beetle, but all that frass has their DNA on it, either because the chewing leaves it on there, or for some boring species, actually that's part of their, they rid themselves of the woody waste through something that they call frass. That's the truth. That's what happens with coconut rhinoceros beetles, for example. Sometimes the pest is so small, you don't really see it with the naked eye. So some of our methods are likely just picking out whole individuals. And then finally with spotted lanternfly, I just laugh all the time in this context, they're the worst criminal ever. They leave their DNA everywhere. So this is just a leaf with honeydew from spotted lanternfly. That's gold for eDNA, right? So any of that's just covered in their DNA. So it doesn't take much in terms of what a spotted lanternfly leaves on a leaf or a side of a tree that we can detect it. So you don't, when you think about how to apply these tools, you don't just kind of go out willy-nilly and look around. It's like, well, what does my species do? Where is it going to leave its DNA? How am I going to go and get it? So from that point of view, we developed a couple different tools that we named. I don't know whether these are great names, but this is what we started to call them. The first one we did actually was on a small vegetable farm in New Hampshire. And this is the first time we tried it. And this is actually a friend of mine's farm. And I realized from just walking through his operation that when they harvest everything from the field, vegetables, they have to wash them, and they have to wash them in a situation in a container that's regularly cleaned, bleach cleaned for USDA purposes. So it should be free of all other DNA every time a new crop goes in. But all that crop comes in, and anything that was living on it and feeding on it or just around it left their DNA. So as soon as that crop goes into that wash water, all that DNA comes off. One of the nice things about surface DNA is that it moves really easily into solution, either through rain or through wash. It comes right off. So now all that water is full of insect DNA, basically, and probably a lot of other. So we figured out if we just take a little bit of that water like it was a stream, and we follow aquatic DNA protocols where you just push that water through a little filter, and then all the DNA is on that filter, and you take it back to the lab, it should work. And it does work. It works really well. We haven't explored it very much since then. So if people are interested in that, I would love to find some places to push that. We used this to find brown marmorated stink bugs in New Hampshire before they were super common. Recently, we've kind of expanded that out for that biosecurity protocol for Capra beetle. In this case, what usually happens in inspection stations is that you pull out some portion of the rice and you look for all the little fragments of Capra beetle or live larvae. What we did is just, instead of searching through that visually, we just took that rice and washed it again, moving any DNA that was on it into solution. Then you take that wash water, that rinsate water, and then you push it through the filter, and you take the filter back to the lab. I'll show you some of those results a little bit later. Now we're kind of scaling up a bit, and we're saying, well, if that works, and we know from some of our other trials that rain will move DNA off of surfaces pretty easily, so why don't we catch the rain? And you can still do that, but we decided that waiting on the rain was a real drag. So what we decided to do instead was to take DI water and put it in what looks like a pesticide container, but those are not pesticides, it's just DI water. We're the rain. We're kind of washing the, in this case, vineyards. And then you have a bucket underneath it that catches the drip water. And then that drip water should be full of DNA. And that's also been very true. So I'll show you some of those results as well. And then finally, we took the idea of a swab and we like supersized it and decided that a regular paint roller can function like a swab. So we disinfect, make sure those you can just buy them at Home Depot, but we UV sterilize them and make sure there's no other DNA on them. And then you just take it out and you look like you're crazy and you're painting the side of the tree, but what you're really doing is the same thing as a swab and the DNA in a murder scene or whatever, is that you are taking that swab and running across the surface, and any DNA that's on the surface gets transferred onto the roller. Then what we do with that roller is we just rinse it like you would at the end of the day when you're done painting your house. You just sort of rinse it out. And then again, we move that water through a filter and take that back and try to find our target species DNA in that water. So we now have like a whole kind of list of things that work, and they work differently for different species, right? So there's not one method to rule them all. There's not one thing that works consistently all the time. So you have to use that natural history knowledge and figure out where and how you're going to find that DNA. So now I'm going to give you a few examples of some of the results we have, just to kind of give a sense of, does it really work? Is it really solving that problem or helping us in terms of the probability of detection when things are very rare or hard to see, or in some cases, it's just dangerous to be out there. There's lots of uses beyond just the abundance piece. So this is what we did in Hawaii. This is for coconut rhinoceros beetles, which are a major pest of palms. Right now it's spreading through the Pacific. What we really don't want is for them to show up in Mexico and Florida and the Caribbean. So this is a worldwide pest that we're kind of worried about. So they're on Hawaii now. So we were out testing some of our methods there. That's one of my research staff members looking crazy, painting the side of a palm tree. That's that roller we just sort of put in the bag of DI water and kind of rinse it off. Then that's the filtering. That's what the filter looks like. There are other filters. We're moving towards some other filters that are cheaper, but that's a good filter. You move the water through that. That captures the DNA that was in the water. Then you take it back to the lab and run it. You can run it in a couple different ways. The easiest way is to use just a standard qPCR. For those of you who are familiar with all that, you just run a qPCR analysis and look for that single species DNA. There's other ways of doing it. So here's just the detection probability, whether or not we're doing 10 samples at a site or 20 samples at a site. They do have conventional pheromone traps there. So, and then we were doing them alongside of those traps. So we were considering the trap results with the eDNA results and seeing how far up we can push detection probability. So you can see we can get detection probability of near 1, like no problem finding them. If we run a trap and we do DNA and we do it in the dry season, which means most of the DNA that the coconut rhinoceros beetles left on the tree is not washed off because of rain. A little bit less good, I guess, when you're doing it in the rainy season because of that rain effect. Both solutions are better than just traps alone. Here's one for it's published, but it's for spotted lanternflies within vineyards. The graph on the right again is detection probability with the number of samples or the number of plots. In this case, a plot is about 3 rows of vineyards, 10 meters long. So that's what we, and this was the rinsing. So that's us with our DI water and our buckets running down the edges of vineyards in New Jersey and trying to collect any eDNA. We were also at the same time recording any visual survey, any visual sightings we had of spotted lanternflies. We did it in 2 times of a season. One when spotted lanternflies are all grouped up, not in a vineyard nearby. And so the vineyard prevalence is really quite low. That it imitates a very low abundance situation. And then a little bit later when they spread out from the forest into the vineyard. The eDNA is the red line and the visual detections are the dash line with the sort of shading representing uncertainty. So clearly the red line is always above the visual. Visual for spotted lanternflies is, we kind of know is not great, but this is a quantification of how not great it is, I guess, compared to eDNA. So you can see even with low prevalence, we can detect them with a 95% certainty. That's what that dash line is, with about 10 samples, 10 plots surveyed. Whereas for visual detections, we barely got above that 95% certainty with 30 plots sampled. And then when they're a little bit more abundant, but still kind of rare, honestly, you don't see them we didn't see them very often in this study. You could confirm that they're there with 95% probability after 5 plots, and then about 3 times that amount for visual surveys. Here's our capra beetle stuff, which was it's just making its way through peer review. This one worked actually so well that we freaked out APHIS. They're a little uncertain. But it's very easy to find eDNA and stored grain from a capra beetle. So here we have the detection probability again. And then at the bottom, what we did was we were using, collaborating with some folks up at Otis, the Otis lab in Massachusetts, is that we spiked some of that grain with known numbers of beetle larvae. So we can kind of convert between number of beetles and kind of the spiked grain. And then the beetles were taken out. So that's why we mean spiked, is that they're not, we let them grow in there for a little while, then we took them all out and then we tested for eDNA there. So we weren't picking up individuals anymore. And the answer there is we get detection we can get detection of capra beetles in about, what, 5 beetles, approximately 5 beetles per 100 grams of clean rice. So a little bit more if you increase the number of qPCR runs that you do. There's some, you should do 3, but you can do 6. That's just because of the stochasticity in terms of the chemistry of the qPCR. You don't want to do it just once. You want to do it 3 times. But those curves go up really fast, right? So the answer there is that it doesn't take much at all in terms of DNA in that rice to be able to, like, one beetle leaves basically enough, one little beetle larvae leaves enough DNA in a pretty good volume of rice for us to detect that they're there pretty readily. One question we always get is, that sounds great, but I'm a little leery of this DNA thing, right? Like, where's the DNA? How long does it last? Are we going to get false positives? Meaning that maybe the critter is not there anymore, or their DNA got moved, but they didn't. So what is the possibility of that? So that's a great question. So there's a couple of things to talk about there. One is that DNA, once it's deposited by an individual, degrades pretty quickly, usually from UV exposure in terrestrial systems or microbial action. So this graph, there's not too much. I just want you to notice that they all go down, right? And they go down really quite quickly. So this is the copy number of DNA from our target organism. This was brown marmorated stink bugs, and how long they're exposed to UV. And that time scale there is about a week and a half. So it starts to get so low that we have a hard time detecting it after about a week. So it doesn't last that long. And the other thing is that, again, we kind of rely on the fact that DNA moves easily through water, and that's true for rain as well. So as soon as you have a rain event, it kind of washes the world clean of the DNA that's there. And so that's another reason why you're unlikely to pick up anything that's about a week, 2 weeks old in a terrestrial system. In terms of movement of DNA from one place to the other, that's a good question. And so I can probably answer some of this, but it's going to depend entirely on the species, honestly, in terms of what's the likelihood of that happening. So, but one more thing is that we include, if you're doing this well, and if you ever work with a group that's doing this, make sure that they have all kinds of internal checks in their process to make sure that they are not moving the DNA from one location to the other. So you have to have a certain level of OCD to make these things work well, and internal checks to make sure that you're not moving from one site where spotted lanternfly is to another where they're not, but you brought the DNA with you. So you want to include all those checks, and you want to ask and make sure if anybody gives you the results. These are called field negatives. Were all your field negatives negative? Because if they weren't, that means you have a contamination issue. So there's ways of gauging how bad that is and when that happens. And what we do, for example, when we work with New York State for spotted lanternflies is, well, first of all, we haven't had any contamination issues. But if we did, then we would know right where they were, and we would just exclude that data. So it kind of gives you a chance of identifying when you might have that happen. One final bit here is that, do you have to do it just for one species at a time? And the answer for terrestrial eDNA, and the answer is no. So people have been using some of these methods that we've developed and some others to do all kinds of terrestrial eDNA sampling. And one thing that we did for insects was just do our tree rolling and our spraying of understory. This is in the Pinelands in New Jersey, and said, what do we find just in general? What kind of arthropods? So this is a figure of those results. And so we pick up, I mean, a ton of different arthropods. So from flies and butterflies and moths, beetles, true bugs, bark lice, bees, wasps, and ants. And then the bottom part is just breaking out the more rare things that we see, from thrips to lacewings to mites, all kinds of things. So we're a bit limited by how much genetic information is available for all these different groups to get the identification all the way down to species. But we clearly pick up, you can do a kind of a good assessment of arthropod diversity, either in a agricultural field, in a forest next to a place where they're spraying, whatever it is. We're kind of exploring how we could use this information within an IPM context, actually, of what kind of in-field management, what impact does that have on the rest of the insect community, including anything that's considered a beneficial insect. So yes, you can elaborate it out. Basically, the bioinformatics and the lab part are a step higher in terms of complexity, but it does work. So are we to the point where we can go out like in Star Trek with a tricorder and tell you everything that's there? No, eDNA still has false negatives. You can't know everything that's out there, but it does reduce those false negative surveys, meaning that you go and look for that and you get zeros. You think it's not there, but it really is there. And that's common when you're looking for really rare things. And it's very common when you're using tools, or the efficiency of your trap is not great. So what eDNA does is it gives us basically a step change in that, and it allows us to reduce those false negatives by a good bit. That's what we've seen so far. But it is not, it's not truth. You know, you can't go out and say if eDNA says it's not there, it's really not there. Few final things I'm going to go through is that we're not the only ones doing this. And in fact, you know, your guy at Cornell, Mark Whitmore, and his lab have been really doing some cool stuff with hemlock woolly adelgid and their biocontrol agents using eDNA. And also at Charlin Partridge at Grand Valley State has been innovating air DNA tools for hemlock woolly adelgid. So that's a whole other thing. If people want to talk about what the heck is air DNA, I'm happy to do that as well. One more thing is that another group, Eva Siskard, who does some great work, but there's some other folks in the US that are doing it, is if you take flowers or leaves, and you dunk them in water instead of spraying them, and then do the same thing that we do and move it through the filter, is that you can detect all kinds of pollinators. So that works remarkably well for flowers and figuring out what has visited those flowers. So that's just a heads up that also exists. And then finally, a big thing for me is making eDNA more accessible and affordable to anybody. And so, you know, having it kind of all contained in the lab, and we've been doing the research pieces of it, but it feels like to me, we're at the point where it's time to simplify and follow aquatic eDNA lead, where they have developed these kits. You just send them out to a citizen or a research agency or whatever, and they don't need lots of things. You have it all there in the kit. They can collect the sample and send it back to you. And then making the other side of that for the mini species looks, either the flowers for the pollinators or in our case for the trees and the pinelands, is how to make it less reliant on a bigger lab, and can you make that sequencing cheaper? And the answer to that is yes, and some really cool new innovations in terms of sequencing. Okay, so I want to acknowledge, like, the funding comes from a lot of different sources. Some jumps off the bridge with us at the very beginning through SARE and that program, because, you know, they're the ones who originally funded some of our work in New Hampshire. Of course, McIntyre Stennis, the USDA, we've been working with the Department of Defense on coconut rhinoceros beetles, there's just a whole group of folks, so I want to acknowledge them. And then, of course, the team that's gotten us here is also kind of broad, and so they've been sort of fantastic folks to work with. Some of them my PhD students, some of them my colleagues, some of them postdocs, some of them undergrads. So I just want to make sure that I acknowledge their contribution to all this as well. So I think we should have plenty of time for questions and discussion. I'm really looking forward to hearing what you guys think. I can stop sharing, or whatever you guys think is the best next step.
Yeah, stop sharing sounds good. Thank you, Julie. That was very exciting. I'm sure there's going to be a lot of questions. So folks, feel free to unmute yourself or type in a question to the chat. And thank you once again, Julie. That was fantastic work. I think folks have the ability to unmute themselves. While maybe folks are marinating on a question, I'm curious also about the false positives, or the potential for that. Do you know, with the segments of DNA, do you require one full length of that segment that is indicative of a particular species? Do you require an intact length of that, or can there be segments that kind of overlap and match within that?
Yeah, so it has to be one full chunk.
Mm-hmm.
And the longer the chunk, the easier it is to know that that's that species and that species alone, just because of probability, and having, you know, the longer chunk means less possibility of random chance that it matches with something else. But one of the false positive piece, like, for example, if you think that chunk might have randomly amplified for some other species, you can double check that by taking, if you have a positive qPCR for that chunk of DNA, you can take that result and send it to the quick and easy Sanger sequencing and get the whole thing, and then just confirm. So we do that a lot with spotted lanternfly and for New York State. if we get a detection eDNA outside of what they're like, really? You know, when you get that response, then we take the qPCR product basically and Sanger sequence it to make absolutely sure it's spotted lanternfly, not something else. And then the final thing I'll say is for aquatic DNA, what they've done, especially with freshwater mussels, because they're a bear, to figure out, because they're so closely related and they have a very strange inheritance of DNA, is they'll develop 2 chunks. And so if the 2 chunks match, then they're very sure, right? So they basically have 2 assays, and if they match, then they're good to go. And if they don't, then they have some question and they dig in a little bit deeper.
Great, thank you. Okay, and I see a question from Emma in the chat. For eDNA and rice, can you get the levels of infestation, or is it a simple yes or no if the species is there? Also, are there thresholds for beetles in rice, or is it rejected if there's any evidence of the beetles present?
So we didn't directly test whether or not the amount of DNA fit to the number of beetles that larvae that we knew were in there. But I will tell you, in general, that's pretty much true. Although there are people who are in eDNA land trying to find a way to take the exact copy number of DNA to go to an exact number of individuals for some species. But on the pretty much universal, I think it's pretty clear that the relative amount of DNA matches the relative abundance that was in there. So you can kind of get a relative sense of what's going on. We didn't do it for rice, but I'm almost certain it holds. The threshold, do you mean a regulatory threshold or a DNA threshold? Or maybe I'll just regulatory. Yeah, for Capra beetles, any evidence is sufficient. That's why we started with Capra beetle, because there's not a threshold number that would send it into regulatory action. It's any evidence, at least in the US. I think those results are quite cool. Like I said, it kind of, APHIS was like, whoa, that's a lot of positive. So we're working and think about what would happen there. And honestly, the thing to do there may be to go to what's called an environmental RNA. So RNA, if you work with it at all, you know that it breaks down very quickly and very easily, but it does persist long enough that you can get it through a filter and do what we would normally do. So what that means then is that if you get a RNA signal in the rice, for example, you know there was something alive there very recently. So that they're developing eRNA techniques in Australia for biosecurity purposes, and they move to RNA for that reason, because they need to know that it was recent. Very recent. So that's still, all this is still worldwide. We're trying to figure out whether or not you should use eDNA or eRNA in that kind of inspection biosecurity realm.
Great. Other questions, feel free to unmute. Otherwise, I have a backup question. But yeah, first, love to hear from other folks in the audience.
I have a quick question.
Sure.
So for using the rice as an example, how long is the turnaround? So if you get a shipment in of rice and you're checking it, how long until you can clear it to go through?
So it depends on what you have set up at the inspection area. So we did qPCR, right, which is a very not sexy version of molecular tools at this point. And in fact, there are small portable units at this point. So if you had it on site, and the way we extract the DNA is from, you can use pretty standard stuff. You could go from a bit of rice that you collected and rinsed out to an answer in about an hour, maybe 2. If it was on site, right?
That's impressive. I had no idea it would be that quick.
Yeah, I mean, the tools, especially with all the multi-species stuff, is pushing all the molecular and the bioinformatics stuff pretty far. Like you have on the front end of that. The qPCR part of eDNA is like no, it's just, if I can do it, anybody can do it. Let me put it that way. It's just not that involved. And then, you know, we're sort of awash with qPCR machines right now because of COVID. because that's how you did all your COVID tests, was with qPCR. And so there's a bazillion of them running around on the secondary market. So we can know how to do it, and it's pretty straightforward.
Great. Maybe one final question before we change things over to more of an informal meet and greet.
Hey, Julie, this is Brian. I just was wondering, you indicated a little bit there with the rice, but just how regulatory officials are using this. This is like new territory for them, and they couldn't necessarily institute a quarantine just based on eDNA, at least at this point. And do you have any insights on that? Or
Yeah, I have a lot of insights on that. So it varies by who you talk to, even in the same agency, right? So New York State has been on board from the beginning. I think that honestly, that's in part because of their experience using eDNA for aquatic invasive species. They've been down that road. They know what the pitfalls are. They know what to think about. And so they weren't that scared. They're like, yeah, let's do it. And so we've been working with them a pretty good amount. They use our information for spotted lanternfly, for example, to do follow-up trapping, increased trapping and doing the physical looking, because of course you can't you still have to have something in hand. You have to have the critter in hand before you can do any of that. So an eDNA detection is not sufficient. So they use us as a broad scale, quick, like we can do a good chunk of New York State in about 2 or 3 weeks. We can do on roadsides where they tend to spread. So we can kind of do a quick run through and say, early warning system, we're getting DNA out here. And then they will follow up with all their work. So they kind of use us as a way to do that.
that seems like a great way to
Yeah, I think that's
zero in.
right now, that's one of the better ways to move forward. Now, aquatic eDNA world has been they're plowing that ground for us right now. So how do they use eDNA tools for aquatic invasive species, for endangered species management? They are working groups with the policy people and the regulatory people and trying to figure out what are you supposed to do with an eDNA detection. So there are some great papers on that I think is going to plow the ground for how we do this with pest insects. There are other folks that usually don't tell me this to my face, but I know it is like they are very skeptical of this, right? And they're reluctant to move to a new method. They're very reluctant to do it. And so that's why, for example, with coconut rhinoceros beetles, I've just sort of said, they're never going to do eDNA on its own. It's always going to happen with a trap. So if that's the case, then let's not pretend like they're happening independently of each other. What does it look like when they happen together? And in particular, for a pheromone-baited trap, right? You're pulling all the beetles in, but that trap doesn't have a particularly good kill rate, right? It's not going to kill everything that comes close to it, but it still moves everybody in. So if we just roll all the trees around it, can we effectively turn that trap into like a super machine of detection by letting the pheromone do its job, and then we do our job of trying to collect the DNA around it? So that's policy-wise, that's another avenue. I'm like, that's another way we probably should explore, is not thinking about them as exclusive use. Is it what happens when we're doing them together?